Well, we can go ahead and get started here as folks continue to join. Hello, everyone. Thanks so much for joining us for the second evening of the Michael J. Bond 75 MD Alumni Undergraduate Research Science Symposium. Tonight's event is titled Cortland and Chemistry, Georges Blemur 96 and Gilead Sciences Incorporated. My name is Rachel Sliversky, for those of you who don't know. I'm a member of the development team, and I'll be serving as a moderator this evening. We do ask that you please mute your computers for the duration of the presentation, but don't worry, we do have an opportunity to ask questions at the end of tonight's remarks. So this event is part 2 of a 3-part virtual series that we're hosting as part of the Science Symposium this fall. Part 1 of the series was last Thursday, October 15th, and featured Dr. Everett Joseph in an event titled An Evening with a Scientist. And then the third part of the series, a Women in STEM panel, will be held on Thursday, November 12th. Things are finalizing, details are being finalized in the coming days, so we'll be sure to send more information as we have it. First and foremost, the faculty committee for the Science Symposium would like to thank Dr. Blemur for his generous support to endow the symposium, as well as Dr. Bill Bartheline for his continued support. I'll ask Dr. Bartheline to say a few words at the conclusion of the program, but Dr. Bond, would you care to say a few words to get us started?
I'm always thrilled to be with faculty, and as this program progresses over the course of the next hour, hour and a half, whatever, I would like the students to really understand the purpose of this, because this is really for you. It's really not for us. And I want the students that you have some of the greatest faculty in the country. The faculty in this country is so dedicated to the student, where in so many other places, the faculty will teach a student class as part of their job. Here, it's their career, and it's a huge difference. So when I went to Cortland here, I was horribly disappointed when I went to upstate medical school because the faculty there, as great doctors they were, and their lectures were outstanding, there wasn't that nurturing education that we got at Cortland. And it just makes you so much better a student, so much better a person, and it opens your eyes as to what you can do. And as we see through the years, we've had outstanding speakers, all Cortland graduates who are doing incredible things with incredible companies all over the country and even the world. And for the students in the audience, that's you in a few years if you do it right now. So study hard, give it everything you got, and use your faculty, their friends, their educators, and a great resource. I'll stop there.
All right, thank you very much. So tonight we'll be hearing from 2 individuals. First and foremost, I want to introduce my co-host this evening, Dr. Frank Rossi, an Associate Professor of Chemistry and Science Symposium Committee member. And Dr. Rossi will be joined by Georges Blemur, our guest this evening. George graduated from Cortland with a BS in Chemistry and Geology after earning his Associate of Arts in Chemical Technology from Broome Community College. George has over 30 years of pharmaceutical experience. Although he started his professional life as a chemist, throughout his career, he has held top positions such as Senior Analytical Chemist, Senior Group Leader of Analytical Development, and Manager of Analytical Development at respective pharmaceutical companies. George now serves as Director of Quality Assurance and Compliance at Gilead Sciences Incorporated in Foster City, California. I had the opportunity to meet with George earlier this year when the world looked a little differently, but we spent about an hour together on the campus of Gilead out in the Bay Area and talked about all things Cortland. So I'm glad that you all have the opportunity to meet him, even if it's virtually. So Dr. Rossi and George, feel free to take the floor.
Great, thanks, Rachel. We kind of prepared a few questions, and Eric actually took our first one. So maybe we can just go back to that. Which is, can you tell us a little bit about what brought you to Cortland and your early experiences?
Well, thank you, Dr. Rossi. And well, let me first like to take this moment to thank you, Rachel, Dr. Rossi, and all of you for inviting me to be part of this year's Science Symposium. I am quite honored to be with you this evening. Now, to go back a bit, I just want to let you know I was born in West Indies, Haiti, which is a Caribbean island I'm sure you are familiar with. Although I was born outside of the US, I was raised in Brooklyn, New York, good old Brooklyn, New York, where I attended elementary school, middle, and high schools. My higher education actually led me to SUNY Binghamton, where I spent a couple of years pursuing my bachelor's degree. However, during those early years, I had also spent time with Brooklyn College and received an associate degree in technology. Now, right at the end of my junior year in Binghamton, I did learn that Cortland was offering at what you would call a 3-2 master's program in teaching. Yeah, that was at the end of my junior year. If I were to enroll prior of my senior year at Binghamton, then I was going to be able to get into this program, and which is what I did, because quite early on, I knew I wanted to teach science as a career. So I had decided to transfer to Cortland during my senior year to pursue the master's program. Now, I don't mind sharing this part of my education story with you, because I truly believe that it is something that your current students should learn from, because I went through it. Once I completed all my required courses and credits for the master's program, unfortunately, I was not able to be awarded a degree. And that's because that I had some dues that is owed to Binghamton. And not student loans, mind you. It's just, dues that I failed to pay while I was in Binghamton before I even joined Cortland. So once I finished my degree, I had to leave Cortland without claiming it officially. However, as luck had it, though, I was able to start my career working in the pharmaceutical industry right after I left Cortland. And I think that was the luckiest thing that could ever happen to me. However, after a couple of years, which is 6 to be exact, I was able to reconcile all my debts with the university, and I claimed a degree at Cortland. However, because that took so long to get back and trying to get that degree or claiming that degree, the curriculum for the Master's in Teaching has changed greatly. And it was a New York curriculum because it was for teaching. They actually had added at least 3 additional education courses to complete the program. By then, I was definitely out of the Cortland area. I was fully working for a pharmaceutical company, and I think I was in down south in South Carolina when I was working there. And therefore, it was impossible for me to stay at Cortland to complete those 3 courses. The school and I went back and forth a little bit about this. And, we agreed that the best they can do for me is just give me the chemistry degree, bachelor's, and leave the MS teaching alone or for another time. So this is really where this was my journey from high school to Cortland. You have another question?
Yeah, no, thanks for sharing this. Can you talk a little bit about what that first job was? I understand that was not really too far from here over in Norwich, and what it was like when you went from school to your first job.
Sure, certainly. Being the fact that I've been in the industry for so long, this is going to seem to be quite a long response to what you just asked. So please bear with me a little bit. I think I've considered myself to be one of those lucky ones who had the opportunity to work within my chosen field. And of course, it is in science, specifically in chemistry, right after college, because there's a lot of people who does not get that opportunity. They usually end up doing something different than what they have studied in school or they have learned in school. I did start teaching secondary school as a teacher, but that did not last long because the position was temporary. It was temporary because I was taking over for a professor that went on sabbatical leave. After he came back, I had to leave, and I think that may be a blessing in disguise because Once I left teaching for about a year or so, I went straight to the pharmaceutical industry. And this is where I consider to be my first career job. And that was a job that was recommended by one of my Cortland professors, because I was really involved with the program at school. I had a lot of professors that I was working with in the laboratory space, in education. So when I had a chance to get a recommendation from this professor, and that was in for Procter & Gamble company in Norwich, New York. And this is my very first time ever working in the industry. I ended up working in a location where it was sample management. I was a new hire, new entry level. And this is where I really started getting my experiences working in Procter & Gamble. Now, during those days, recommendations from school professors were highly recommended, and also they were respected. And I'm sure it is the same today for your young graduates, that, especially for students that come up straight out of school, because you don't have any experience to speak of. So they really look at the work that you do undergrad, any laboratory work you were doing outside of the classes that you were taking. I was lucky enough that I was also a lab assistant, if you will. So I used that as an experience from the school, plus the recommendation, and I was able to get that particular job. And really, this is where the door was open for me to actually continue working in the pharmaceutical industry. Now I can go ahead and continue telling you that after I didn't stay too long at Norwich. After a few years of getting a couple of experiences under my belt, working in the laboratory areas, I actually had an opportunity to go all the way down south, first time ever, in a New York guy who grew up in New York, never been in the South before. And here I am. I was working for a recruiter, really, which is where they have those independent recruiters that just got a hold of me and decided, wanted me to know if I want to go to GSK, GlaxoSmithKline, but it wasn't GlaxoSmithKline then. When I first started, it was a SmithKline Beecham, which is not in existence anymore because now it becomes GSK. So I've decided, no problem. It was for a chemist, a position on working in a laboratory. That wasn't a step up for me after the sample management gig out in Norwich, Eaton. So I decided that I'm going to move from Binghamton and go all the way to South Carolina. And lo and behold, I did receive the position working as a lab chemist. Again, it's really important to know that around this time around is that the industry was not hiring in a manufacturing plant anyway. They were not hiring like professional chemists or folks that has 4 years or 6 or even doctorate chemists. Around that time, they were hiring technical folks, 2-year folks, associate degree folks to work in the laboratories. So when I was asked to come in and join that particular company, I was one of the few chemists they were hiring. So this is where not only I was building my experience in the laboratory, but also get a taste for managing, because automatically I became in the management field, working with these technical 2-year degree individuals that were already in working in the laboratory. So I stayed there for a couple of years, working in that laboratory, what you call a quality control laboratory. That plant was manufacturing products such as over-the-counter medicines like NICE, Geritol. I guess if you guys remember those days, those products, where it was not development work, it was more commercial product that was already out there on the shelves. So I was in the laboratory performing testing associated with those programs, with those products. I stayed there for a couple of years. Then I've asked for a transfer to go back to New Jersey, at least close to where my family was at. And they had gave me that transfer. So I moved on to another plant doing working in the laboratory again, testing toothpaste, if you can believe that. testing toothpaste. Very simple process in testing, using HPLC, GCs, all that good stuff. And of course, as I'm going through this, I'm just gaining experiences and working in these laboratories. And later on, I've started to become more serious about this career. Then I went and looked for a position out a company called Sharing Plow in New Jersey. This is where I started working in the development areas. Sharing Plow, which is now Merck. Sharing Plow is not in existence anymore. Merck has acquired it. But I worked there for a couple of years in the development space, working with methods, developing methods along with other researchers. Then I spent a couple of years there. There is a trend to that, isn't it, right? I moved to my very real management role came in down in North Carolina. After a few years out in with Sharing Plow, I moved down to North Carolina and worked in a company that is not really what you consider to be big pharma. Big pharma is the company I just mentioned to you, GSK, Merck, Sharing Plow. This company was called a CMO, which is what you call a contract manufacturing site. These were the time where big pharma were switching their business model, where they would support other companies' product and doing the testing, manufacturing and testing of this product and release them to, so they would to the sponsor, for example, to a company like Merck. So I was asked to go to this company in North Carolina and became a supervisor for a couple of different quality control laboratories because it was high throughput testing. They need multiple labs to support those testing. Again, those testing were already established. The methods were already established. It was routine testing. It's just a matter of running the methods and generate results for these products and then release the product to market. So that's what you call a CMO, one of those manufacturing organizations. Now, after a few years of that, I was in that site for about 15 years. Between the time I started with them as a supervisor, I moved to a managerial role in the quality control laboratory. So now I have a lot more labs to manage. And then after a few years of that, maybe 6-7 years, then they moved me to what you would call a development lab, research and development laboratory within that same facility. This is where I will look at methods, redesign those methods, or create new methods to support any new program that's coming into the site. And so it wasn't really quality control, but it was more an area where you'll revalidate methods or redevelop methods to support any new program that's coming from these other companies. And I stayed there for about 4 or 5 years. So in total, I was in that site for about 15 years, 13, 15 years, I believe. And after a while, I kind of got tired of working in the laboratory. It's not tired per se, it's just more like I gained all the experience I can get working in the laboratory space. And I wanted to use all the experience I've had. By now, I had over 25 years of experience working in the laboratory. I was looking for a way to get out of the laboratory and do something different with my experiences. And lo and behold comes Gilead Sciences. And the only reason why I was able to make that connection with Gilead Sciences is because they were one of my clients while I was working in that particular CMO. And we worked on a project together, and they had liked the work that I did, my team did, and so on and so forth. And somehow they heard that I was ready to make a move. And so they contacted me. And they told me that to come over in the West Coast. I'm like, never been to California before, it's the first time. And I thought, hey, you know what? Why not? So I was very excited about that. So I came to to Gilead Sciences at a time where they had decided that 70% of their testing activities and manufacturing activities will be done externally. So they want to focus more on the development of programs and their therapeutics. They were extending their therapeutics areas. So they would not want to spend a lot of time manufacturing or testing product that's already on the market, or even certain late stages in clinical phases. They want to transfer all that outside to outside vendors. And my job is to create this network of laboratories with my experience in quality control and development experience. They thought that I'll be a good to be able to lead a team of people to bring in new suppliers into their network that they can contract the work out, the testing out, the manufacturing out to them. And once we build that network, then we could manage your sites for Gilead. And that's what I did. I came in, and it was a very satisfying role. I did travel all over the world, picking out different labs, see for the best fit of labs for the programs that we had. I spent a couple of years doing that. We have labs now from Germany. We have labs in India. We have labs in all over Europe. Of course, we have a lot of labs in the United States because of the effort that I and my team had put forward to build that network. After about 5 years, we have built the network. We have over 50 to 60 labs and manufacturing plants out there that's supporting Gilead for its multiple programs that are either on the market or the clinical at a clinical stage right now. And there's an opportunity that comes up to actually now audit those manufacturers, the suppliers, the labs, and everything else. And they thought that it was a good idea to actually go into this role as a director in the compliance and audits division to be able to go ahead and audit these sites for compliance and either compliance or in compliance. So that's what I'm doing right now. It's been about a year since I moved into this role, and I have a great team of people working with me, and the program is growing, and I'm really excited about the next few years coming up because we're going to make some changes, and so hopefully it will come out the best. But right now with the COVID-19, we had to make some changes. But at this point right now, we just doing the best we can to make sure that we are within a regulatory compliance with these sites.
So as I think many people know, Gilead has a long history of developing antivirals for treatment of HIV and Hep C. And then you've really been at the forefront with remdesivir and antivirals related to COVID. Can you talk a little bit about how life has changed for you over the past 9 months? Maybe even a little bit about when you first kind of got a sense of what was happening with COVID at Gilead.
Yeah, sure, I can do that. Since March is when the pandemic has started. Okay. Since then, Gilead, recognizing the situation earlier on, I'm going to break it into two separate discussion here. What we have done since the pandemic, and also touch base on the COVID-19 and how we got there. In February, March, we are noticing that we had noticed that there was an increase in cases of this virus, and once we realized that this virus is upon us, and our researchers actually recognized that we had one product, a nucleotide analog that we had used for Ebola, seems to resemble very closely to what COVID-19 looks like. And that was very important because these antiviral drugs, they are all similar. They are all similar in the sense of their structure and their the way they will attack any virus in the system, in your system. So, but the only problem is with Ebola is that we went through the whole phases of Ebola about 10 years ago when it was rampant in West Africa. Unfortunately, on our last phase of trial, we realized that although it was safe to take and use, but the efficacy was not there. There was not much of a big difference between what just normal controls in the clinical setting versus taking that particular drug. So we have shelved it. We have shelved it and put it away. But in the process, we had produced a lot of that material. So that material went to storage until COVID-19, when we realized that, hey, you know what, we might be successful with this candidate because it is so aligned with what we have. So those folks who knows much more than I do about this start working on it and realizing that, yes, there is a chance we can help fight this virus by the way it attacks the virus while it's in the body. Unfortunately, we cannot prevent somebody from being infected. We can only treat it, but the virus itself had to go into the cell of the human body for it to actually take hold. It works in a way that it's amazing. But the way it works is that I don't know if you guys know the whole structure and how the virus infected healthy cells. But once it goes into the cell, and it has to go into the cell to actually replicate, our drug candidate, what it does, or remdesivir, if you will, what it does, it goes into the when the code to duplicate the virus, it actually interfere with that process, with that mechanism. So it will not only has to start replicating, but it could stop it from replicating any further into in these healthy cells. So by knowing that, our first thought was, let's look at the worst cases. If we're going to put in any effort, if we're going to work hard in trying to save certain patients, let's look at the worst case possible. And that's what we did. The first trial that comes out and the phase 3 trial that comes out, we're focusing on those patients because then later on, look at other trials for less severe patients. But unfortunately, the worst patients can only be saved by a small amount. So the efficacy of that drug did not go too well with the worst patients. However, when you go back and look at less severe patients, where folks that may be in low oxygen flow, people that may be in ventilators a little bit, we have a much more success. And that's where we spend most of our trials and studies. And I think the NIH had come in, which is the gold standard of trials, came back and show that we're able to reduce hospitalization of patients from 14 days to 10 days, so 4 days less than what would have if we did not have remdesivir around. So of course, and then with that knowledge, the FDA had given us the approval to distribute that drug under emergency use, or at first it was for under compassionate use. However, as the trials comes in and they see more data, then they allow us to distribute it to under emergency use. Emergency use only mean people that are hospitalized and they are under ventilators, and that's almost like the last resort for a particular patient. So we're able to do that. Of course, as we go through this, we're looking at trials. We're working on and increasing our production facilities and network to be able to distribute these drugs across the board to everyone once, hopefully, we get the approval from the FDA. I like to say, I think somebody mentioned just recently that just a few hours ago, we have gotten that approval in the US to distribute this drug across everyone. So now there's no barrier to anyone but that patient has to be in hospital. They have to be under low flow of oxygen, So there's a few requirements before they can get the drug, but at least there's no barrier to any patient. Any patient right now can take that drug. So which is a great accomplishment for us, for all of us who's been working so hard trying to get this drug to the patient as soon as possible. Right now, most of us are working from home. About 10 to 50% of us are working on site. Those folks are needed to be in the laboratory. These are development folks, but a person like myself, where the role does not require to be in a building or in a laboratory, a good number of us are working at home since March, and that's what I've been doing for the past 6 months.
Great, that's good to hear all the details of what's going on. Was wondering if you had any advice to our current students in terms of them thinking about their futures. So many of them do come from different backgrounds. We have many students that come from two-year colleges before they get to Cortland, and then others, of course, that start here as freshmen. Some are very committed to one thing and they end up going that way, and then others will take turns like you did. I just wondered if you would kind of give some advice to our current students.
Sure. One thing I can tell you guys is that anything you can take away from this journey I just explained to you is, I think I touched base a little bit, that you need to have a great relationship with your faculty, professors, and teachers, and learn as much as you can from them during the time that you're at school. Because I believe that helps me a lot, because as I mentioned earlier, is that when I transferred over to Cortland, I don't know, because maybe I had a couple of years of school in my belt, so I was pulled in into the laboratory, working along with some great professors in their research. So that really helps me a lot. And if you're not doing it for a chemistry individual, you want to have that under your belt. I don't know if there is any opportunity to work in the laboratory, being a lab assistant, or even anybody who's putting chemicals on the shelves. All this will work, will be better served when you finish school, because right now, the industry, the companies, especially in development, in laboratory space, are looking for somebody that has at least some sense of how to actually work in the laboratory. Having those kind of experiences does help. Also, networking. Right now, it's much easier than when I was in school, with LinkedIn, social media, and all that good stuff. I think that helps a lot. And also, don't be afraid to choose or to take on a position that may not be the best position for you at the time that you start. Like I said, when I first started, I didn't go straight into the laboratory. Actually, I was in a room, and all I did was just managing samples, sending samples from different places. Although I was in the lab proper, but I was never given the opportunity to work in the laboratory, because that was not the reason why I was there. But I did take it anyway, and I make it work for the best of my advantage. So it's important that you stay focused on what you want, because also is the fact that there are many opportunities where I was given to move away from the laboratory, from the science, and go and do manufacturing, for example, because a lot of time you're gonna see that you're gonna be overlapping with your experience, and some folks might think that, hey, you might be a better suited for this role. But if you want to stay in science, and there's great opportunities out there right now, to get into the laboratories, to work as even as a quality assurance QA in the pharmaceutical industry, laboratory QC, lab analyst, as a chemist, and you don't have to have a master's or a PhD degree to do those positions. Your experience, if you allow yourself to do so, will get you to where you want to go. For example, 30 years ago, 25 years ago, I was not expecting to be a director for compliance and audits, right? And nor was I expected to be a manager for development laboratories. But the experience that you gain as you go along helps a lot. And I'll be honest with you, when I first started, I didn't think I was going to do far because I didn't have the PhD because of what was my goal. My goal was to work in the industry and go as high as I can. But at that time, people were saying that, well, you need the PhD, you need the master's, and so on and so forth. But I just keep on trucking along, get my experiences, move from one company to the next, as you can tell. And I think that helps a lot. I'm not saying to not to be loyal to a company you work for, because loyalty is where you are at any given moment. However, I find that if you want to reach where you want to go, you may want to take some chances. You may want to leave a comfortable role and go in somewhere else. And I think I did that throughout my career. Like I said, I move all around. I've started from New York. I went to South Carolina, Georgia. I came to New Jersey. I went to North Carolina, and look where I am today. I'm in on the West Coast. So if you start and you want to stay in one company for the long haul, I'm not sure you'll be as successful as I am because of the move I've made. Now, it may not be for everyone. I was able to move because I had that luxury and that flexibility to do so. And of course, as I move along, I gather experiences, and also experience is good. Not just one type of experience, but multiple type of experiences. And so when you are ready to graduate, at least have something to start off with. Like I said, recommendation is good, but classes only may not make it. You have to have some physical experience working in a laboratory. Even if you're going to be trained, when you get to the laboratory, that's something that's important. That you will get trained in how to run instruments, how to operate certain highly skilled, highly expensive instruments, HPLC for example, gas chromatograph, IRs, moisture analyzers, anything that could help you get that leg up while you're undergrad can only help you. So one class that I now remember that I really love, and I think that class really gets me going, is the equipment instrument, analytical instrument class I had taken for a full year, and I've learned a lot, and that included high performance liquid chromatography, gas chromatography. So when I first sit down for my interview for my very first job as a lab chemist, I remember talking to my hiring manager, explaining to them that, yeah, that's what I was able to do that as an undergrad in Cortland. And I was able to list out all the necessary parts of that instrument and the acquisition system that was used to integrate peaks and all that good stuff. So that was impressive for a person who never had any experience in the lab before. And that's how I got hired. So do your homework, learn as much as you can when it comes to instruments, because once you know the instruments, everything else will come into place. Because we all here to measure. And if you don't know the instrument you're going to be working with to measure, it doesn't matter what sample you're going to be using, what type of, if it's a liquid powder, that doesn't matter. It's the instrument. So focus on that instrument and take that with you, and it will help you along the way.
Frank, I'm going to break in. One of the things that Georges doesn't know is this very expanded undergraduate research program we have. We have 5 faculty on here. You ought to explain to him just how robust our undergraduate research program. I think he would be very pleased, whether it's you or Peter or Teresa or Legion or Joshua. You're all doing undergraduate research. So someone should share with him just how robust it is today.
Does someone want to take that other than me? So we get a little bit of other voices.
I'm happy to share my experience. I had a student, Christina Gutches. She started in 2011 when I actually also started working at Cortland. She worked in my lab for 2 years. Just like George is saying, she initially was just washing some water bottles since we were doing a lot of water sampling. And then later, a semester later, she started to work on the instrument and running the ion photography. She loved it. After Cortland, she went to Syracuse University. Because of the research experience, she came in actually as an education major, but she realized she really enjoyed doing research and went to SU. Wanted to do a master just to kind of see how the grad school will work out for her. A year later, she emailed me back saying, Dr. Jing, well, I said, call me Lily. I really enjoy research. I'm not gonna just do a master, I wanna do a PhD. So she basically switched to PhD program after 1 year at SU, finished her PhD work in 5 years, and got a postdoc position at Yale.
That's wonderful.
Yeah. I
was share a story with one of my former students who also went to Yale. I guess that'll be our theme. He has PhD at Yale, and I talked to him last night actually, and he's at Pfizer now. He's at the competition, and he's been there about a year, and he's getting ready to do his first real big scale up. So he's in process research, and so in process research, you're scaling things up from the laboratory, and he's getting ready to run this reaction where the starting material is $2 million worth of starting material. And he's understandably nervous because you can't screw up. And it took I think a year to make this starting material, and it's going to be a $2 million experiment. And he's like, oh no, it's going to be done before Thanksgiving, but I'm nervous about it. It can't go wrong.
Yeah. That's another thing too, because people doesn't realize how much money it goes into developing a drug and process chemistry. And I know this because when I was building my network of contract laboratories, a lot of these materials are coming from the process lab, and it's very expensive because part of keeping managing these, having this oversight for these labs was that recognizing that they have to handle these materials in a way that they cannot just throw that material away or abuse that material or spend in a way that Gilead would not want, because it's very expensive, it's very hard to produce. So and the people that actually involved in making that material are people coming straight out of college, because even though we may have researchers that we may have a program director or a researcher that might be responsible for that material, but the people that actually on the bench that actually producing this material are people who are coming right straight out of college at Cortland, and but they are expensive. You know, they are expensive, and there's protocols to follow and to make sure that you don't throw the company's money away, because they could be in the $1 million range. So yeah. So by the way, whoever mentioned about the undergrad program at Cortland, that's wonderful to hear. Like I said, I mentioned to you guys that as students, start now. I don't know if you guys register in LinkedIn. You'll be surprised how much information you can get from LinkedIn if you are targeting a particular company, Pfizer, Gilead, or Genentech, or Merck, one of those huge companies. There's a lot of information in there for you to start doing your homework and targeting folks that work within those industries, within those companies. And they sometimes send you a response. I get that all the time. I get people that some people find that I was working at Gilead and some of the effort we are doing towards COVID-19. I was getting a lot of questions, and how do I get into Gilead and so on and so forth. I do respond sometimes, depending on where that person is coming from New York especially. I'll respond to that. So I'm sure a lot of my colleagues doing the same thing. So take advantage of what you currently have today. Like I said, I didn't have that kind of, I had to work really hard and build a bit of a network. And if you're lucky enough, you might get somebody to give you a phone call, but it's much easier these days. When I'm hiring people right now, I'm able to find those folks pretty quickly because of the LinkedIn as well as all the social media. And of course, you also have these independent contractors, what we call headhunters. And that's their job. They do this on a full-time basis. And it doesn't matter how many years of experience you have. If you can come out straight out of college, and they will work with you. They will find a place for you. That was just something that started when I graduated. And right now it's a full industry. So there's a lot out there that you can use. And if you want to take a break from school, you don't have to go straight to master's. Of course, it's not for everyone. Having a PhD and working in the laboratory, as long you are passionate about certain things that might be important, like maybe a particular therapeutic areas that you want to get involved in. Yes, I would advise you to have a PhD from the get-go. But right now, after 30 years in the industry, I have PhD folks working for me for the past few years. And but they came out straight out of college. They spent X number of years in school and getting that degree. And of course, that needs to be respected. And it's great. You don't have to go back to school again. But there are those that started school and finished school, started to work, and then they take a break and go back and do their master's or PhD. And then they come back and turn around and come back to the company. So there's many options out there. So it's not just one straight path. There's multiple ways you can be successful in the industry.
Thanks, George. We're actually going to, before we do any other questions, we'll go ahead and have Dr. Bill Bartheline, who's on the call, say a couple words this evening. Bill, if you can hear us, you're on.
I don't know. My problem has been if you can hear me.
We can hear you.
Okay. So I'm here.
Yeah, opening the floor to you to say a few words this evening.
We've had significant difficulties for me to get here tonight, so I'm not able to say that right now.
Okay, well, thanks so much for tuning on. Dr. Bond or Eric, do you have anything?
Yeah, I do have a question for Georges. You know, you're in quality assurance and the nation is waiting for the vaccines. What is your level of confidence? Because, you know, usually it takes years to bring something from research to market to testing. This warp speed is extraordinary. What is your level of confidence? Georges, you're on mute. Oh, there you go.
Okay, and I can understand your concern, but I'm going to give you, I think we don't talk a lot about this in public, but this drug did not just came out of the blue. We had this drug for 10 years. The whole purpose of that drug was to fight Ebola. And we went through the phase 1, phase 2, phase 3 of that drug about 8-10 years ago. So it was just sitting on the shelf. So the time that you are thinking about for a drug to develop and to market, we did that. We just got lucky. If I'm not even sure it's lucky because so many people are getting sick and die from this thing, but lucky enough that we did not have to go through the phase 1, phase 2s, and the early late phases of this drug because it's already taken care of. We already had shown safety profile for this drug about a few years earlier. So all we had to do just go ahead and go straight to the last phase to show efficacy for this new virus. So that's what we did. That's what we spent. That's where the 9 months. Now it was still a challenge for us because we knew that this drug was going to come to market. So where we spent a lot of time beside initial clinical trials to show that it will work, we spent a lot of time expanding our network of the logistical network to get this drug into market. And that's what a good number of our colleagues spent time on. So there's great confidence that drug will work, has worked, and the data is showing that. And I think the last trial that came out shown that we were able to reduce the death rate by 70% just by this drug alone. And that's a big deal. I know we don't tout it, we don't celebrate in public the accomplishment, but a lot of us has been spent a lot of time. I personally, being the fact that I was in the middle of trying to making sure that the sites that we know that's going to manufacture this drug all over the world have to be qualified to perform the task. My team and I were working diligently trying to qualify these sites, and on top of COVID, it was not an easy task. So I'm talking to you right now, but it was a trying time, and everyone were involved in trying. They know exactly what we're trying, what we're working hard for, and so having this approval today, it's really amazing. It's really amazing of the work that we went through to get where we are today. And I don't know about you guys. I don't know if you know about Gilead. Gilead is not the type that flaunted their names all over. We really work in the background. The only reason why you know about this is the fact that we're on TV all the time. Our CEO was talking to the newscast, to the news outlets. He even went to the White House. But if you go into the campus, and Rachel could attest to that, you will never know that this is a Gilead campus because there's no big name, big banner in front of the campus. The only place you'll see a Gilead signage is in the buildings. But it's really, when I first moved into this area and searching for Gilead, the actual campus, I was expecting to see this arch of Gilead names everywhere. No, it wasn't. So it's just the company culture. That's the way it is. But we knew this is important. So we had to put everyone, it was an all hands effort. So to answer your question, yes, the product is we have great confidence in the product because we knew what the data looked like, but we had to show new data because of the COVID-19. And that's why we went straight to the NIH to show the world that whatever data you're going to come back with is going to be very positive. And if you can show that, then everyone else will accept it as opposed to just our own trial data. And and that's where we are today. And that's how we're able to get that approval today. And also getting that emergency approval to provide it to patients that were on their last breath in the hospital.
Okay, one of the things I'd like to say is one of the things I really enjoyed you talking about was that from college to where you ended up, even though it's in chemistry, is still not a straight line. And for the students, you need to know that when you're going through your coursework, we all say at one time, why do we have to know this? Because it doesn't seem to be that related. It's very difficult. I found out with anatomy when they even had names for places that didn't exist in the body, they named it. But you really have to know things. And as you're going along, the better you're going to be at what sometimes is unrelated in your mind that ends up being related somewhere down the road. But some of the skills you pick up, again, will be helpful in your career. Knowledge is growing. And you want your knowledge to continue to grow. You want it to continue to expand your horizons. And you never know what road it's going to take you down. I mean, when I went to medical school, I went to college first to do marine biology and to show how well advanced I was. I go to a school that had no water anywhere near it, and I want to start marine biology. So, it's the science. I knew I wanted science. I knew I wanted to go far. And then it was just meeting great faculty who kind of directed you into things and helped you develop your interests, and more importantly, helped you to do things that will promote your interests. And that's what Cortland is all about. So I did 4 years of independent research in psychology because we had a great psychology professor who at the time, this is 1971, was doing a lot of independent student research, and it wasn't really that big at that time. So I ended up spending 4 years with him. My dear friend, Wayne Marley, who also went to medical school with me, he was a chemistry major, and he was doing the psychology. So we learned a lot in that psychology, as did Dr. Berger in the medicine of things. So just want to surround yourself with quality people who are interested in learning, who are people that you could learn a great deal from. They're wonderful people to be with. And if you could do those things, you'll always do well.
I do want to mention that I know I spent a lot of time talking about chemistry, but we do have a lot of other science discipline in the lab, in the industry as well. Biology, microbiology. I mean, we take all comers because of the therapeutic areas that we are covering. So it's not just chemistry. Chemistry is good, but we also converted a lot of biologists into the laboratory as well after a couple of years. So we take all comers. Thank you very much. I do want to mention that if you guys, again, I don't know if does the school still host recruiters from industries? When I used to be in college, they tend to do that as well. Is that still going on?
Nan, you need to tell them about Handshake and all the things that we do.
I will. Hi, Georges. I'm Nan Pasquarello. I'm the Director of Career Services. And yes, we do still host recruiters. Right now, due to COVID-19, everything is virtual for a little while. But as you were speaking, I looked up Gilead Sciences in our Handshake platform and noticed that we are connected already to your company. And I noticed you have a couple of jobs that are advertised. So students, log into Handshake, check out Gilead Sciences. And we're also promoting LinkedIn and the networking that you brought up. So I'm really glad to hear you promoting students get experiences, network, go to recruiting events, and so happy to hear everything you had to say this evening.
Thank you very much. It could be lengthy, but I was trying to get everything I come out of my head, so everybody can at least share the ideas, everything that that they need. Because, like I said, when I was an undergrad, I did not know how my life was going to turn out to be. I just knew that I need to graduate. So after graduation, now what? You know, I could either have gone to higher learning or go through the industry, and that's why I chose. I went straight to the industry because I see opportunities. I just take it, and then here I am today.
Thanks again, George, for joining us. And thank you to Dr. Rossi for helping to host this evening. And thank you to everyone else for participating in part 2 of the Michael J. Bond 75 MD Alumni Undergraduate Research Science Symposium. As I mentioned earlier, more details to come about our Women in STEM panel in the upcoming weeks. And thanks again for joining us, everyone. Have a good evening.
Yeah, thank you, everyone. This was wonderful. Absolutely wonderful.
Thanks very much. So long.